All right, good evening, everyone, and welcome to Genomics Lite. This is our third in our infectious disease series, and this evening we're being joined by Nicole Wheeler, and we'll be looking at some of the big questions in infectious disease research. So before we hand over to our speaker, I'll introduce myself. So I'm Em, I'm the Education and Learning Officer for the Wellcome Genome Campus. We've also got Fran sort of hiding behind the scenes on the call today, and we're sort of here to help make sure everything runs smoothly. In case you've not used webinar before rather than like a regular Zoom meeting, you've spotted that you can't use your microphone or your camera, but there's some other ways you can interact with us or ask us questions if you need to. So in the sort of bottom right hand corner of the screen, there should be a chat box function. If you've got any general questions or general issues, like you can't get something working, that's where to ask us any questions there. We've also got a dedicated Q&A function. It should be at the bottom of your screen as like two little speech bubbles. If you've got any questions for Nicole specifically about her work or anything she's talking about, if you can pop those in the Q&A, it makes it a lot easier for us to find them and ask them to Nicole. We've also got some polls, so they're like little multiple choice questions that pop up on the screen when we need them. I know if you're on mobile, sometimes they don't pop up, so you can just use the chat box if you can't find any of the other things going on. Before we hand over to Nicole, I'm going to give a little intro to the campus and the work that we do, and then I'll pass over to Nicole. So let me share my screen again. So this is the third in our series on infectious diseases, and next week will be our final session in this series, and we've got a career pathway panel to celebrate International Day of Girls and Women in STEM. So we'll be joined by 4 different researchers looking at how they got into research positions, so that'll be really interesting as well, and we'll put some information in the chat box about how to sign up. So this is the Wellcome Genome campus itself, in case anyone hasn't heard of it or seen it before. We're based just outside of Cambridge in England, and it houses a lot of different science research on site. At this point, I like to see who is furthest away from campus. I currently live up here in Scotland, but Nicole, I think, might win by default, and I'll let her tell you why in just a second. But if you can find the chat box and let us know where you are from today, like where you're joining us from, it'd be really fun to see how far away some of you are. I got one from Hong Kong. That might be one of our furthest. UAE even, that's pretty far. Canada, that's pretty far. Quite a lot from the UK, so hello to all of our UK joiners. Quite a few from America. Awesome, so we've got quite a mix today. Oh, one from Glasgow, fellow Scot. We've got quite a mix of people today, but I think Nicole might still win as furthest from campus. So on the Wellcome Genome campus, we've got lots of different research groups, but the 2 biggest research institutes are the Wellcome Sanger Institute and EMBL EBI. EMBL EBI is all to do with data science, so all of the staff there work with data, whereas the Wellcome Sanger Institute is a bit more of a mix of data and what we might traditionally think of as like wet lab biology. Nicole used to be based at the Wellcome Sanger Institute and is working somewhere else at the moment, and again, I won't spoil her talk too much. Everything to do on the campus is to do with genome. That's why it's in our name. And the genomics is the study of the genome, which we would call the organism's complete set of genetic instructions. I'm going to put a poll up in just a second asking you how you would rate knowledge of genomics. This isn't like a test. It doesn't matter how you would rate your knowledge. It's just to help us see who's in the room without seeing you. So I'm going to put a poll up. It should just jump up on your screen. And it's a little multiple choice thing that you can fill in and just gives us an idea of how much people in the audience already know. Awesome, quite a mix. So we've got quite a lot of people that know a fair bit, some that know a little bit less, and that's all awesome. That's why we're here today, to find out a little bit more about different areas of genomics. Awesome. So on the campus, there are 6 main areas of study: human genetics, cancer, cellular genetics, parasites and microbes, the tree of life, which is looking at biodiversity, and biodata. So today's talk falls sort of between parasites and microbes and biodata, and we'll find out more in just a second. Before I hand over to Nicole, I just wanted to point out some really nice resources that might be particularly useful for anybody studying for GCSE or A-level or any other sort of biology exams. We've got our Your Genome website, which has loads of articles and like quizzes and facts about different types to different parts of genomics. And you can sign up for our education update as well if you want to find out more about education events like this. You can also get in touch with us by email or on social media as well, and I'll stick all these links in the chat box in just a second. I think that's everything from me, so I will stop sharing my screen and I will pass over to Nicole. And as I said, if you've got any questions as we go, stick them in either the chat or the Q&A, and we'll ask Nicole some questions throughout the talk. So Nicole, I'll pass over to you.
Thanks, Em, and hi everyone. Thanks for showing up to the talk. Just to break the mystery about where I am, I'm currently in New Zealand, staying with my parents and escaping COVID. So I'm almost exactly on the opposite end of the world right now. So I'll just share my screen. There we go. Hopefully you can all see that. Great. Yeah, so I will be giving a talk about some of the big questions in infectious diseases that I've been grappling with in my career. I currently work as a data scientist and a bioinformatician, and I'm consulting for the Nuclear Threat Initiative at the moment. So before I launch into the talk, I thought I'd talk a little bit briefly about my career, in case you guys are wondering what to do with your lives and where to go and how to plan a career. So I started my career in New Zealand. I decided to do a bachelor's in biochemistry because I had no idea what job I wanted. I knew I thought science was cool and human health was important. And basically biochemistry gave me a lot of options later on once I'd made up my mind. So my undergraduate, some of the things that I learned was that I thought microbes are really cool, and I thought genomics was as well. And I mostly learned at this stage through going to lectures, talking to lecturers who are really passionate about stuff, and reading a lot of popular science books. So that gave me enough direction to choose to do a PhD in biochemistry, where at that stage I knew I wanted to do genomics and infectious diseases. And I learned in my PhD a lot more about bacterial genomics. I started developing software and I learned about machine learning. And in the PhD stage, a lot of this was teaching myself. I had a question, I didn't know how to answer it. My university was kind of small and didn't have many people who worked in my field. So I just looked online and learned to code and things like that. I also went to seminars and conferences where you get to learn a bit more about sort of the research that's going on today that you might not find in books. Then fortunately out of that project, I got a postdoc at the Sanger Institute in the UK. At that stage, I was still set on genomics and infectious disease, but I'd also developed a real love of AI or artificial intelligence. So I wanted that in my career. And at the Sanger Institute, where we produce a huge amount of genomic data, I learned how to work with big data. And I learned a lot more about genomics and global health issues as well. There's a lot more focus in the UK and at Sanger on what's going on in the rest of the world in terms of infectious disease. And at that stage, I was still learning by myself, but I also got to meet lots of cool colleagues over at Sanger and go to talks to learn more about what I could do and where I could go. Later, I took a job as a data scientist. Here, I wanted to bring in some global health work into my career and do some more applied work, sort of developing tools for middle and low income countries who might want to do genomics and better understand infectious disease. So there I learned about challenges faced by middle and low income countries and the challenges in translating science into something you might use in the real world. And here I really learned just from meeting people through my job and trying to apply science. And then finally, I recently left that job to take a consultancy job at the Nuclear Threat Initiative. The name is a bit misleading. They don't just do nuclear threats anymore. They also do biosecurity. So my job there is to develop tools for improving biosecurity around the world. And that was really because I wanted to move into a role that let me have more real world impact. So some of the things I'm learning in this role is that the really technical genomics-y science stuff is just a small part of the solution to infectious diseases. and that in the real world, incentives matter in terms of getting science done. If you build a really fancy tool that's really cutting edge, but it's expensive and it's slow to use, people won't use it. So you've really got to think about the real people who are going to be using your solutions. And here I've really learned from talking to lots of people working in biosecurity and in industry, and all the things that they've learned throughout their careers. So maybe some take homes from my career so far. A bioinformatician career path didn't exist at my university when I left high school. We didn't have any data science career paths either. So I couldn't really have planned ahead for the career that I've ended up in. Each job that I've had has taught me something a bit new and changed my direction in my career a little bit. And the way I sort of meandered through my career so far is to develop skills based on what I seem to be good at, what I thought was really interesting and what I enjoyed, and also what people needed at the time. I originally started out in the lab, but every job I had, people kept wanting somebody who knew about bioinformatics and computers. So that's where I ended up. It helped that I found it really interesting as well. So now my career really focuses on infectious diseases. I think in terms of human health, this is a really exciting area to be working in, partly because infectious diseases are able to behave so unpredictably compared to other human diseases. As we're all well aware now, a new infectious disease can suddenly spring up and spread around the world before we really know what we're dealing with. And once it's spread, it can adapt over time in response to our attempts to eliminate it. And one thing that's a really big problem across infectious diseases is growing resistance to treatment. So things like antibiotic resistance. And while it's really easy at the moment to picture the impact of infectious disease in the form of outbreaks and pandemics, infectious diseases really have a far-reaching impact on all of modern medicine. A lot of advances like much safer childbirth, invasive surgery and chemotherapy really rely on our ability to prevent and treat infections. And if we lost that ability through antimicrobial resistance, we'd really have to rethink the risks posed by these different practices. And modern medicine could change quite fundamentally. So what are we doing about infectious disease and antimicrobial resistance? One of the things we really hope to work on is developing new treatments for infectious diseases. But new treatments are hard to come by, and it can be really hard to get them to market. So we're also trying to develop better recommendations around responsible antimicrobial use to try and preserve the treatments that we have today. We're also working on better communication and collaboration between hospitals and countries, because we recognise that infectious diseases can hop borders all the time. And so if you only know what's going on in your own hospitals in your own countries, you're bound to be taken by surprise. So a really cooperative, collaborative, global approach is key with infectious diseases. And also we're working on better surveillance to notice outbreaks as they're occurring, and to head off looming threats. And genomics is becoming a really important part of this, partly because the cost of genome sequencing has dropped so dramatically over time, meaning we can really use it in a wider range of cases and at a bigger scale. So I wanted to ask you guys what you think we could use genomics for in the fight against infectious diseases. So if you could put some ideas in the chat, that would be really good.
So there's been a lot in the news around sort of how we use genomics in infectious disease. So I'll be interested to see what comes up. So we've got a couple, vaccines is the big one coming up at the moment, probably no surprise given everything in the news. Identifying new strains of a virus, PCR, following a path of disease, like how it spreads and evolves, identifying people who might be more at risks, finding out maybe more about the structure of a virus. Lots of great ideas coming up in the chat.
Fantastic. Someone asked what Moore's law is from the previous slide. This is a theory about improving computational power over time. So computers used to be really big and really clunky and not many people could use them. But over time, the computers are getting faster and faster, which means more people can have them and they can do more things. So that line is showing the improvement of computing power over time. And you can see that genomics is improving at a far steeper rate than computers are. So while we think our advances in computing are extraordinary, we've made real leaps and bounds in genomics, actually. So these are some of the ideas that I came up with about what we could do with genomics. This is not an all-encompassing list. There are heaps of things we can do. But some of the things you might be interested in is identifying what pathogen someone is infected with. One of the ways you might decide whether someone who's looking ill has the flu or SARS-CoV-2 is with genomics. You can also identify different strains that came up. So within a particular disease like COVID or SARS-CoV-2, there are different variants. Some might be associated with different outcomes, as we've seen with the recent strains that appear to be more transmissible. You can also do surveillance to see what's going on in your community and around the world. For instance, some people are looking for genetic material of SARS-CoV-2 in wastewater to see the general load of the virus in people's wastewater, and also what the major strains are across the community without having to sample every person in that community. And lots of people picked out that you can develop vaccines with genomics. So these days we're able to prime the human body with bits of genetic material from a virus that we're interested in to help the human body fight off an entire virus. So that's a really exciting new development. So there are some big questions that sort of loom over our fight against infectious disease as we're trying to do our day-to-day technical work. Some of the ones that I've been thinking about a lot are how can we help everyone in the world fight infectious disease? How can artificial intelligence help us fight infectious disease? And also, how can we handle the risks associated with all of the technologies that we're pushing forward to develop? So firstly, how can we help everyone in the world fight infectious diseases, not just people in the UK? So the UK has done extraordinarily well in using genomics to help understand the spread of SARS-CoV-2. This is an overview, which is very complicated, of all of the data that we have been collecting using genomics on SARS-CoV-2 in the UK. I won't go into detail explaining the whole figure, but suffice to say, we've done an enormous amount of genome sequencing. That's allowed us to piece together a family tree of the virus, which you can see on the top right here. And you can see in the bar plot at the bottom, all the samples that we've sequenced over the course of the pandemic. So you can see a small bump around sort of April, which was that first peak. And then you can see another one coming up in around November, and then another one quite recently. One of the things you guys noted about how useful genomics is the ability to identify different strains of the virus. So we've been talking about some more highly transmissible strains that have come up. In the more recent part of the bar chart, you can see some really fat pink bars and some really fat green bars. Those are two of the really transmissible strains which have really taken over what we're seeing in the genomic data quite recently. And you can see compared to March and April, we've got lots of colours stacked on top of each other, which is lots of different strains. There's a real change in the mix of virus that we're seeing over time. So that's something that we know with genomics, which would have been hard to identify otherwise. But while the UK is doing exceptionally well in producing genomic data, when you look at the SARS-CoV-2 genomes that have been gathered from around the world, there's a real difference in volume of sequencing. And most of the data is coming in from a small number of quite wealthy countries, predominantly the UK and the US. So that means that there are a lot of countries that have a lot less information to work with when they're trying to design infection control strategies and understand what's going on with COVID where they live.
Nicole,
so one of
we've got a really good question about what determines whether a virus is a different strain of the same virus or a different virus altogether. Like, where do you draw the line between it being a completely new virus?
That's a really good question. I think even strains people struggle with a bit. So you can see, I think in the data, we've got this thing on lineages. We've got B 1.177 and 1.1.7. These are all different IDs that have been given to different variants of the virus. Basically what we're doing when we pick out different strains and different species of organisms is we look at the genetic diversity And we try to draw some lines based on maybe a cluster of genetic variants that look quite similar, and then hopefully a distinct cluster. So in the tree over here, you can see the new variant in the green is a really good example. We've got this big brown cluster with other colors mixed in, and then a very distinct green bit. So that is relatively easy to call a new strain. Other times it's not as easy. It can be quite arbitrary, to be honest. We just try to look for a jump in genetic diversity, really. And to say something is an entirely different virus is usually to say something is a new species. This is really hotly debated. Basically, we look at a tree and we draw a line where we think something looks different. But often people will disagree on that. And sometimes what we think is a completely new species might not look so new when we gather more genomic data and we see that there's a lot of genetic diversity joining two species, which is very common. So calling something different strains or different species is kind of a convenience term to help us associate different qualities with what we're looking at. But it can be a little bit arbitrary how we draw those lines. So we're interested in democratizing genomic sequencing, which means allowing more people to have access to it and having broader representation of genomics around the world. At the moment, we're facing some opportunities and some challenges in this area. One of the big opportunities has been things like Zoom and other video conferencing software, which means that you can gather a group of people from around the world and have a conversation without needing to get on a plane and all meet up in the same place or get on the phone. So you can share your screen, you can be in the lab with someone else on the other side of the world who's also in the lab. It means there's much more opportunities for people around the world to work together and share knowledge. Genomics has been getting a lot cheaper and a lot more portable. That means you can use it in a wider range of circumstances. You don't need a big institute like the Sanger to do genome sequencing. You can do it out in the field. And also companies, more companies are popping up to provide different services around genomics, which means that if you're a small team and you have some skills, but there are other things that you're lacking, you can send part of the process away to the company to have them do it for you, which means it's much easier to get into genomics in the first place. But there are still challenges here. One of the big ones is if you're working with infectious diseases, you're often going to be working with human samples. And to work with human material, you probably need ethics approval. Whether you're a big institute or a small group, ethics is something that you always need to consider. And so that can really slow down the type of sequencing you can do. And you need to be able to navigate the ethics approval system. So that's kind of a non-negotiable. Also, if you want to do a good job with genomics, there's some training that you probably need. You want to plan which samples you take well, you want to collect them properly, you want to pull out the DNA properly, and you want to analyze that data well. So there's some training that you need, but more of that is available online or through Zoom, and so it's more accessible today than it was in the past. And also funding and resources. Genome sequencing, while it's getting cheaper, it does cost money. You do need to buy equipment. And so getting access to that is still a challenge. We've had some really cool successes in recent years. One is around portable sequencing. So there's a group that, well, there's several groups that are developing kind of lab in a suitcase models where you can pack all the equipment that you would need to do genome sequencing, including in the top right image of these photos, we've got MinION sequencers plugged into laptops. So these are tiny sequencers that you can take on a plane, you can take out in the field, really handy for doing genomics. So now you can pack everything you need into some suitcases and just go to somewhere in the world and do an investigation, which is what people did in the Ebola epidemic, which feels like a very long time ago now, to help track the spread of the virus in almost real time. So that was a big change in how we do genomics and investigate outbreaks. Something that I was involved in at the Sanger is called the Global Health Research Unit for Genomic Surveillance of AMR. So this was an investment from the UK in setting up infrastructure and training in Colombia, Nigeria, India, and the Philippines. So this was an investment in some bigger, beefier sequencing equipment and capacity in other countries, so that we can get a better idea of what's going on with antimicrobial resistance around the world. And the reason the UK is investing in this sort of stuff is because antimicrobial resistance doesn't just appear in the UK, it can be imported from overseas. So if you want to fight infectious disease, you really need sort of a web of surveillance around the world. It's not enough to just do it in your own country. So this sort of effort is really important to help us really understand what's going on with infectious disease in AMR. Another thing that I've been involved in, which is a bit more kind of grassroots, is something called Project Accessible Genomics. So this is a really small community project that started with some people in the Philippines. It's sort of researchers and people from startups and people from hospitals in the Philippines who've connected with people around the world to set up a really low cost entry-level genomics project for sequencing of SARS-CoV-2. So a lot of this is people volunteering their time and crowdsourcing funding. And so we're putting together a guide of how we got the funding and the equipment, how we figured out how to do the sequencing, so that other people in low-income settings can do SARS-CoV-2 genome sequencing as cheaply and as easily as possible. So another question I've been looking at is how can artificial intelligence help us in fighting infectious disease? Democratizing genome sequencing is great, but we're already producing a lot of genomic data, and as more people start producing more data, the amount of it is going to be quite overwhelming. So we need effective ways of pulling out valuable information from that data, and that's something that artificial intelligence could help us with. So I'd love to get your thoughts on what you think artificial intelligence could do to help us fight infectious diseases.
So again, if you've got any ideas about how AI might be able to help in terms of genomics and infectious disease, pop them in the chat box. One interesting idea around pattern spotting, which I think is a skill that AI has more generally. Looking for patterns where infections might arise, tracking new strains and being able to process large quantities of data. Yeah, I imagine that'd be able to do it a lot faster than people. Predict future outbreaks to sort of help look at the rate of a spread. Yeah, and then a few ideas around it being be more precise than humans or have less errors than humans, or maybe being able to predict possible treatments as well. So quite a lot of interesting ideas coming up.
Yeah, those are really good. So again, this is not an all-inclusive list, but some of the things that I have been thinking about is modeling the spread of infectious diseases across time and space. So this can use genomic data, but it can also use things like social media activity, patterns of people's movement in terms of sort of local movement and flight patterns. It can be looking at electronic health records. It can be using contact tracing apps, which have come up more frequently now. So there's all sorts of different data that we could use to help us better understand how an infectious disease might spread, which can help us plan interventions. Sort of in line with that, we can also use AI to help us detect or predict outbreaks. So at the moment, I think we are aware that there can be a delay between when an outbreak happens and when we realise it and when that information is communicated broadly. And that delay has a real cost in terms of the number of people who might be infected. So AI could serve as a part of an early warning system to start noticing trends maybe in medical data or in what people are posting on social media or what's coming out in wastewater to help us see when there might be an outbreak earlier. Some things that are a bit more patient-centered, you can use genomics and AI potentially to predict which infections might be resistant to treatment and maybe to design the ideal treatment for a particular infection. And also for a given strain, you might be able to predict the strain's ability to cause severe disease. So it might help you, when a patient comes into the clinic, better decide how much attention and care to give them and what might be some higher risk decisions that you might be making about them. So while AI has a lot of potential and it's very good at spotting patterns in data, working with DNA data can be really challenging, both for humans and for AI. So a lot of the progress that's been made in AI recently is looking at image data, because images are really good for pulling out different kind of shapes and patterns and textures, things that the AI can then use to associate with, clinical outcomes for dogs and cats or something like that. But DNA data is really just very long strings of 4 letters. And so there's a bit less to work with there. And so I've got a bit of DNA on the screen here. This is some DNA from salmonella. I have worked on salmonella for most of my career, and I could not tell you what this DNA is. If you didn't tell me it was salmonella, I wouldn't know it was salmonella. I don't know where the genes are in this. I don't know what this DNA does. So even a human expert has trouble looking straight at this data and knowing what it is. So you really need to think cleverly about how we process this information to help the AI out in finding anything valuable in it. Another complication in working with infectious disease is what you're trying to predict is really complicated. So ideally, if you wanted to look at a piece of DNA and say, is this DNA from something dangerous? There's a lot of information you would need to piece together. You probably want to find the genes in that DNA and predict what they do. And then you'd want to know how those genes interact. And then on top of that, you'd want to look at the DNA from a whole organism and use that to predict how the organism is going to behave, what it's capable of doing. And finally, you want to understand how the DNA from that organism might interact with other organisms. So a really nice example is thinking about how the diversity of SARS-CoV-2 and all the different strains that are out there might interact with the diversity of humans to cause different clinical outcomes. That's a really complicated problem, and currently people can't do that, and AI isn't going to be very good at doing it either, just because it's there are so many questions packed into that question of which viruses are going to cause bad outcomes in which people. But there are some things that we can do with AI today. Most of them are around making predictions on DNA based on things that we've seen in the past. So if we look at the piece of DNA and we say, what is it similar to? Some of the things that we could answer are, what species is it likely to have come from? We can do that pretty well. Does this piece of DNA have a gene in it that's similar to other genes that we've seen before and we know the function of quite well? And finally, within our piece of DNA or within the DNA of an organism, do we see mutations and other genetic changes that we've seen before that seem to be associated with something like drug resistance or bad infection outcomes? So this is kind of where we are at the moment with AI. These are some of the things that we're working on predicting. Sometimes it's really accurate, other times it struggles a bit, but this is kind of the edge on which we're working and making progress at the moment. So while that's not predicting biology from scratch, there's still a lot of valuable information we can get from that. One of the projects that I've worked on has been looking at salmonella. And among salmonella, a lot of strains are capable of causing food poisoning. They're quite common around the world, and while food poisoning is really bad, it's not really the worst of salmonella. Other types of salmonella can cause things like typhoid fever. So really dangerous, life-threatening infections. And one of the things we learned when we looked at the genomes of different salmonella is that the ones that cause really severe disease seem to show signs that their genome has broken down. So they have lost a lot of abilities that salmonella that cause food poisoning have. And they seem to keep losing the same abilities every time they adapt to causing severe infection. So that's something that AI can learn and can search for in a lot of genomic data. So this is an example of a dashboard where we've got DNA from salmonella around the world. We've screened it for signs that its genome is breaking down in a particular way. And then we can assign a risk score, which tells us which strains might be more dangerous and which ones we maybe want to pay more attention to. So this is kind of what we could do today. So finally, I wanted to talk a little bit about the potential risks that we are grappling with today and possibly creating as a result of developing these technologies. So I've talked a lot about genome sequencing, and it's definitely something that we do a lot of at the Sanger. That involves a lot of taking DNA from out in the world and reading it. But these days, one of the things that we can also do with DNA is write it and create it from scratch. So that means taking all of the bases that make up DNA and putting it together in a sequence that we want. When we first developed this technology, because DNA is kind of fragile, we can only put together short stretches of DNA, but even that became incredibly useful for different types of diagnostics and other technologies. But because there's such a demand for being able to create DNA, we're getting better and better at producing longer strands over time. And there's a lot of cool stuff that you could do with a long piece of DNA. Some groups have actually managed to write the genome of a virus from scratch and then kind of activate it so it becomes infectious. Some of the other things we can do is edit the DNA of organisms or put bits of DNA in to give them new or different functions. So this sort of technology shows huge promise for things like biotechnology, conservation, environmentalism, medicine. Some of the things that you could do is sort of engineer bacteria to produce medicines or biofuels. So a lot of really exciting implications of this technology. And it's just improving and growing in scale over time. And one really cool thing is that while you used to have to do this in the lab, which took a lot of skill, now you can just write a piece of DNA that you want, send it off in an online order, and then the DNA will be shipped to you. So that makes this really accessible. But while that's really cool, there are also potential risks associated with this. So this synthetic DNA could be thought of as a dual-use technology. So dual-use technologies are things that present a lot of potential benefits to society, but also present the opportunity to do harm. And one of the things that I think about a lot is that I'm developing all this AI to try and figure out what changes in the DNA of an infectious disease could make it more dangerous. As we get better and better at understanding that, we're producing a lot of information that could be misused in the wrong hands. So this isn't the first technology that we have developed that comes with risks. There are a lot of other ones around there. So we can borrow some ideas from other dual-use technologies when we think about how we might want to approach the risk posed by this one. So one of the really famous ones is nuclear technologies. Great for sort of energy production and a lot of exciting advances. But also nuclear weapons are a huge potential threat to humanity. So they're really something that we want to get a handle on. So some of the ways that we've tried to do this is really tightly restricting who can work in nuclear. So it's hard to get jobs, it's hard to obtain materials, and even some information is very closely guarded. We also have agreements between countries about what sorts of technologies and materials will be developed to try and limit the risk of misuse. So that's one example. But another example is cyber technology, so things like hacking. While it's relatively easy to control people's access to certain nuclear materials, anyone with a computer and an internet connection could become a hacker. All the information you need is online, so you need a different approach. So some of the things you might use there are standards within the hacker community about what's acceptable and what's not. And some companies offer rewards. So if you find a vulnerability in their software and you tell them, rather than exploiting that vulnerability, they might give you money or they might offer you a job to help them fix that problem. Another one that's a lot newer is the risks around artificial intelligence. So here we're thinking about potential military applications, but also potential risks associated with AI that doesn't perform very well. So thinking about things like self-driving cars that put people at risk, or medical devices that make the wrong decisions. So some of the things we're developing there are regulatory guidelines about how to tell if an AI is safe and fair, and also a lot of public accountability. So if a company does something bad in the AI space, the media might report on them, and that could be really bad for business. So when we're thinking about synthetic DNA, there are a lot of approaches we could take, but we still haven't really got a clear answer on what the right approach is. So that's something that we're looking at the moment. So when you're trying to decide how to manage a risk, one of the things you might think about is soft versus hard power. So an example of hard power in controlling a risk would be something like setting a new law. Laws can be really helpful in the sense that they set clear rules about what's okay and what's not. And you can enforce strong consequences if people break those rules, like giving somebody jail time or giving them a fine. But laws take a long time to make, and you have to make them all around the world. So that can take years, which if you've got a really quickly developing technology could mean that the law is always years behind the technology and therefore doesn't really cover all of the scenarios that you're worried about. On the other end of the spectrum, in terms of soft power, you can have things like community standards and public accountability. These can change moment to moment, so they're really responsive to new changes, but you have less control over the consequences there. If there's a media report that comes out about bad company behaviour, if there's a lot of news, it might go unnoticed, or if somebody really influential picks up on it, the consequences for that company could be really out of proportion to what they actually did. So what are we doing around synthetic DNA at the moment? People have been discussing what to do about this risk really seriously for about 15 years. In that time, some guidelines have been developed to talk about general ideas about how we might make the industry safe. But it's up to individual companies to take those general ideas about don't ship dangerous DNA to dangerous people and turn those into actions that they do every day. And these actions typically cost companies a lot of time and money. And the choice of actions might vary a lot across companies, depending on how many staff they have to devote, how much time and how much money they have. And this gives customers an opportunity to kind of shop around different companies and find ones that are a bit more lenient if they want to order something dangerous. So one of the things I'm thinking about is, what would a good standard for screening of orders in this industry look like? So the things you'd want to ask is the customer who's ordering the DNA safe to work with? Could they potentially have nefarious intents? Do they know how to work with DNA? Do they have a safe laboratory? Things like that. You also want to know whether the DNA is safe to work with. So does the DNA pose any potential risk? As I said in the AI section, we still don't know a lot about that, so it's kind of a hard decision to make. And finally, what are the special circumstances associated with each order? So there are a lot of really good reasons to work with potentially dangerous DNA. If you want to develop a vaccine or a treatment, that might be something you need to do. So we need to design flexibility in what people can order and why. One really hard question is, what do you do with what you don't know? So there's some DNA that we're pretty sure is quite bad, things like toxins. And there's other bits of DNA that are found all across life. They perform really basic functions and they're probably okay. And then there's all the rest of the DNA that we're not really sure about. Maybe we don't know what it does. Maybe potential risks associated with it are kind of in a really niche context. So what do we do with all the stuff that we're not sure about? If we're too cautious, that really limits who can work with what DNA, and it might cost companies a lot of money. So it might be that we develop a screening technique and nobody uses it. But if we're too relaxed, we might increase the risk of intentional accidental misuse of DNA. So figuring out how to deal with uncertainty is a really big challenge. Another challenge is figuring out how you gather information about what DNA is dangerous. So there's going to be a lot of DNA being ordered or showing up out in the world. And within that DNA may be nestled something that's dangerous. To identify that dangerous DNA, you have to take all the knowledge of biology and danger and gather it in one place to do this check. How do you do that? One way is to take an open approach where you create a public resource. Anyone can contribute what they know about the dangers of DNA. And that would mean that if someone wanted to put together a dangerous piece of DNA, they would have to outthink everyone else who contributed to that resource. But it also creates a really convenient list of dangers that someone might misuse, and it could create a risk in itself. But the flip side of that is the convenience of that list could mean that a lot more people are able to develop treatments and surveillance techniques and diagnostics around that DNA. Another approach would be to use a closed approach, where you develop this information resource in secret, and most people aren't allowed to look at it. So people, if they find something dangerous, need to contribute this knowledge to the resource and then not talk about it ever again. So if I'm doing my AI research and I find something really interesting, I would submit it to this resource and then I wouldn't publish it. I wouldn't tell the media. I would keep it to myself. A challenge there is in figuring out why scientists would do that when it's not very good for their careers. But the benefit, of course, is that we're not creating a big list of dangers that any outsider could look at. But along with that, we might have fewer people working on treatments and prevention measures around those dangers. So I'd love to get your input on what you think. Is the best way to gather this information a completely open approach or a completely closed approach, or somewhere in between?
Awesome. I've just popped a poll up that's got sort of 5 options. So a completely open option, a sort of somewhat open option, but with some restrictions, a mix of open or closed, somewhat closed with very strict restrictions, or completely closed. And yeah, there is no answer to this. So it'll be really interesting to see what you guys think. Awesome. We've got lots of votes coming in. Very mixed. Only a few people think it should be either completely open or completely closed. Most people are somewhere in the middle. The majority right now is actually on the sort of somewhat open but with restrictions to access side, so more open than not. But a decent amount of people also think it should be mainly closed with very, very strict restrictions to access, which is really interesting.
Cool. Yeah, it is very helpful to get your input because this is a big decision to make. And it affects the public. So we want to know what you think.
Yeah, at this point, the majority of people, so 40% of people have said that they think somewhat open with some restrictions would be their sort of preferred way to go, but still very split amongst the sort of different options.
I think that's probably true amongst the scientific community as well. So that could look like something which is a database that only people working in medical research or something associated with risk could access, but a random member of the public, they don't need that information.
Then one interesting question of whether you could use AI to track what people have done with the data, which comes quite like full circle, use AI to track AI. That's an interesting idea.
That's something I'm very interested in. But one of the issues there is privacy. Is it okay to track individual people and what they're doing? So this sort of area leads to some broader questions around if you, whatever job you have, wherever you're working, identify a risk to humanity, what's safer, telling everyone and getting sort of the world to develop responses and prevention measures to this risk, or keeping it to yourself so that you don't share that information around and increase the risk of somebody misusing it? And also, who's responsible for identifying and preventing risks? Is it scientists and companies who maybe create these technologies? Is it the government? Is it something above the government that's international? Who is responsible, and how do we sort of incentivize people to be thinking about this during their work? So if you find these questions interesting, there are a few things that you could do to get more information. One is a docuseries on Netflix at the moment called Unnatural Selection. This looks a lot at DNA editing and looks more from the perspective of human DNA editing and raises some really interesting ethical questions. And also the Nuclear Threat Initiative has an education centre with lots of information about potential risks and the ways people are thinking about addressing them. So with that, I'd like to say thank you for attending and listening, and I'm really happy to take your questions.
Awesome, thanks very much for that, Nicole. I've just popped the links for the 2 things you mentioned in the chat so everyone can find them. We've had some really, really excellent questions in the Q&A, so we'll sort of try and get through a few of them in our sort of next 10 minutes. One that I think links quite closely to the sort of situation at the moment, a question on how close are we to developing a surveillance, a global monitoring system for epidemic outbreaks using like real-time AI?
I think we have the capability. It's about getting it implemented. I think individual countries at the moment are looking at ways of using genomics and AI to monitor infectious diseases. But it's sort of researchers who are developing these ideas, the government who needs to approve the use of those ideas, companies who probably need to get involved. So there's a cool initiative in Australia at the moment that's seeking funding, which would be an AI surveillance system for AMR, which looks across like farms, people, wastewater to see what's out there in terms of antibiotic resistance and how it's changing over time. That's a really good prototype for what we're thinking of. But it takes a lot of funding and a lot of permissions to be able to gather all of that data and process it and then deliver it to the people who need to make decisions. So it really takes an investment from the government.
Awesome. Someone's asked what exactly sort of dangerous DNA is, and there's another question that links to that of like what would be a sort of worst case scenario if someone who shouldn't get access to this sort of so-called dangerous DNA, what would be the sort of worst case scenario from that access?
Yeah, so I can partly answer that. Dangerous DNA could take a lot of forms, and I think it's really up to human discovery and creativity to think about what that could be. Some of the things that are sort of ideas at the moment is around infectious diseases. There have been programs in the past around biological weapons that tried really hard to make really dangerous strains of bacteria and viruses. They were limited to what they could find in nature and what they could sort of manipulate in the lab, which meant that they weren't wildly successful. But with technologies we have today, capabilities in that space would be really different. So we have biological weapons conventions now, which means that if a country is caught doing that, it's a really big deal. But it's still a possibility that that could happen. And so it's really important to design ways of detecting that activity. Another one, which is way more speculative, is around human DNA editing. So we now have the technology to change a human genome. If you found the ability of editing genomes at scale, that could be huge. It could really change who we are as people and our future. So it's further off in terms of how you would do that, but that would be a really scary prospect, I think.
There's been a fair few questions around like getting into bioinformatics and getting into data science. And I think a few people are interested in like the sort of coding aspect. If anyone in our audience was like interested in learning a bit of coding or getting involved in a bit of bioinformatics, what would your sort of best recommendation for sort of looking into it be?
I think coding and data science, I had to teach myself during my PhD, and it's relatively accessible. There are some really good courses online that help teach you the basics of coding. There are some really good data science resources. I'll share a link in the chat for a website called Kaggle. That was one of the places that I went to learn. It's got heaps of big data sets and other people sharing their code of how they analyze it. And it has some nice tutorials and things like that. So that was one of the main resources that I used to learn. I think schools are getting a lot better. I'm learning that people are learning Python in high school now. We did not have that when I was in high school. We had computing, which is like how to format a Word document. So there's a lot more opportunities out there. And I think the coding community are really good in putting together online tutorials that are really helpful. Bioinformatics is a little bit more challenging. I think we're getting better because everyone has to learn themselves. And if you don't learn well, you don't produce great science. But yeah, I think it's harder to get into. It's probably a lot easier to do a university course in it at the moment and really talk to someone who's experienced.
Great, thanks for that link. So if anyone's interested in like uptaking a bit of coding, then definitely check out that website there. There's a nice question
as well. Sorry, just to say that there are also apprenticeships that offer bioinformatics options. So I know that the Anglia Ruskin University in Cambridge have offered actual bioinformatics apprenticeships as well. So you work on the job, but then get a day study release as well. So that they won't be the only organisation doing that. So that is another option. So you get paid as you learned as well, which is another nice option.
Awesome. There was a question that linked to a little bit about what you were talking about at the start about trying to democratize genomic sequencing. And so someone's asked a nice question that, again, doesn't have a specific answer, but I think your thoughts will be interesting. On what extent do you think competition and like political friction between countries provide, like, challenges the ability to democratize and, you know, make globally available genomic sequencing?
I think, coming from the research perspective, I think there is kind of a real motive. If you're in a high income country and you're in a competitive field, to sort of get your name on a lot of papers. And so if someone from a low income country asks you for help, you might say, yeah, I'll help you, but as long as, you know, I get to run the study and all of that. I think there's been a real push for, if a study is being done on a country, for a lot of the scientists and medical professionals from that country to be given credit on that study and to be given capacity to continue that work. So it's not okay for them to just send samples overseas. Maybe you should actually invest in their lab instead. So I think that's sort of a changing attitude towards sequencing around the world, and a growing recognition that it's not okay to just do one study. It's much better to set up something long term, so that people can benefit from that research over time, and they don't lose that capacity once the study is over.
Yeah, and I think we're seeing that at the moment, right? That being able to globally cooperate to fight infectious disease is going to just become more and more important. A slightly different question, but I think might be interesting. Do you think genomics and AI might be able to be used in relation to climate change? I don't know if you've got any experience on that, but
it's definitely not a key area of my expertise. I think there are expectations that as the climate changes, different sorts of microbiomes change as well. So it could be an indicator of change. I think there are probably sort of biotechnological solutions to aspects of climate change. One is around sort of producing more sustainable biofuels. So that's one way that you can use genomics to engineer organisms to produce more sustainable fuels. But I think it's probably going to be one aspect of a bigger solution.
I was on mute there. We're coming sort of up to the end of our time here, and I had spotted a nice question earlier that I think might be a good place to finish off. Someone had asked, what part of the future of AI, or I guess your research field in general, are you most excited about?
I think one thing that I've become very passionate about is bias in AI and fairness. So a lot of AI is built using big data sets from a small group of people or a small number of countries. And that means that it doesn't always perform well across different demographic groups. And we've seen some really bad examples of biased AI that disadvantages certain people. So this year I've been really involved in trying to impact governance of AI, which means if you are a health system looking to adopt AI, how do you tell that it doesn't disadvantage some of your patients? I think there's been a real backlash against unfair AI. So I'm very excited about all of the different attempts to develop fairer approaches and more equitable access to these sorts of technologies. And it's something I'm going to be chasing after this year as much as I can.
Excellent. Yeah, someone sort of followed up on that, saying surely the AI is just as biased as the creators, and I think that's part of the issue, is if only one type of people are developing the code, then it's going to be more biased to that sort of type of people. And I think what you're talking about earlier as well, in that the UK has been really good at doing genomic sampling of like the coronavirus, but then if that's all the information we have on coronavirus, we might be missing out on other information from other parts of the world. So yeah, the more people that can get involved in sort of coding and that sort of thing in the future will only make it better. That is perfectly 5:30, so we are going to wrap up there. I know that there are some more questions in the chat. If anyone wants their questions answered, I'll put our email up at the end and you can get in touch with us there. There's also quite a lot of questions sort of around careers and about getting into science. Our session next week, we will be joined by 4 researchers, and a few of them are bioinformaticians as well. So if you're interested in looking at sort of career paths and talking more about what subjects to study to get into different university career paths, I'd really recommend coming along to that next week. And I'll put all the information for that in the chat in just a second as well. But yeah, thank thank you very much, Nicole, for joining us at such an early time in the New Zealand day. Thank you very much, everyone, for coming along, and hopefully we'll see some of you next week as well.
Thank you, everyone.